Good morning, students. I welcome you back to school after the lockdown. In MIC 432, Food Microbiology, I had taught mushroom cultivation and farming and also chemical preservation of foods. So I'm going to continue with physical methods of food preservation. Physical methods of food preservation, as the name implies, are methods in which physical treatments are used to inhibit, destroy, or remove the undesirable microorganisms without involving chemical processes, antimicrobial agents, or products of microbial metabolism as the preservation factors. There are several types of these physical methods of food preservation. Number one is physical dehydration process. The dehydration process is an ancient method of food preservation. It has been used from time immemorial. It actually involves the removal of water from food because, as we know, an aqueous medium will support microbial growth. So water is a major factor contributing to spoilage or deterioration of food. However, we should understand that it is not the total moisture content that results in the food spoilage, but rather the availability of water that determines how long the food will remain without spoilage. What I'm trying to say is that a proportion of water in the food, of the total water in the food, is strongly bound to specific sites. This available water is measured by what we call water activity. Water activity is the ratio of vapor pressure of water in the food to the vapor pressure of pure water at the same temperature. We know that certain foods that are fresh and raw may have high moisture content. Example is fruits or vegetable. These are food that have high moisture content and the water activity level may be up to 0.98 or higher. So preserving such foods will involve dehydrating such food such that microbial growth is limited or inhibited by the removal of water as much as possible. In essence, water required for growth of the microorganism is removed and therefore water activity is reduced. Technologies for physical dehydration of foods include drying, freeze drying, that is rapidly freezing food and then subjecting it to high vacuum, and then freeze concentration, that has to do with concentrating liquid products by freezing the water content and then removing the so formed ice crystals from the food system. These processes will reduce the water activity of both the food and the microbial cells to a level that is insufficient for microbial growth, thereby inhibiting microbial growth. Due to chemical composition and water binding capacity of components, various foods may have very different moisture content even at the same water activity. You should note that the water activity requirements of various microorganisms also vary significantly. For instance, research has shown that gram-negative bacteria have higher water activity requirements for their growth. Other physical dehydration processes I've mentioned are the freezing and the freeze drying and the freeze concentration. They are all parts of physical dehydration processes. Now, the second method under physical preservation methods are cool storage or frozen storage. Cool storage means refrigeration, while frozen storage means freezing. The third one is controlled atmosphere storage. This controlled atmosphere storage refers to continuously controlling the gas atmosphere in a packaged food. For instance, we have oxygen, carbon dioxide, and nitrogen being controlled and regulated. The advantage of this method is that it can inhibit browning of cut surfaces of fruits and vegetables, and then the flavor of the food is retained. Another advantage is that there's reduction in deterioration or decay of such foods, and the nutritional values are maintained, such as vitamin C. Such nutritional values retained in the food as a result of the controlled atmosphere storage. However, disadvantages include the fact that they are quite expensive. You need to control temperature regularly, and then also different gas composition for different types of commodity are required. Special training also may be required for whoever is operating the process and even the operation itself. Finally, when the pack is opened, it leaks out the gas and then the benefits are lost. Another factor or another method amongst these physical preservation methods is the modified atmosphere storage. This modified atmosphere refers to a gas composition that is modified initially and then changes dynamically depending on two factors. Number one, the permeability of the film, and then the storage structure surrounding the produce. There are two types of this modified atmosphere storage. We have the active and the passive. In the active modification, the atmosphere is modified by pulling a slight vacuum and replacing the packaging atmosphere within the desired gas mixture. In the passive modification, the modification is achieved by respiration of commodity within that package, and it depends on the characteristic of that commodity and the packaging film. Now, another method of physical preservation of food is vacuum packaging. Vacuum packaging removes air from the package prior to sealing. This method involves manually or automatically removing the air from the package, placing items in a plastic film package, and then removing air from the inside and sealing the package. The advantage of this is that it removes the oxygen and thereby inhibiting all aerobic bacteria, aerobic fungi, and then these organisms are not able to grow and cause spoilage. Another method of physical preservation is the various forms of heat treatment. Example is pasteurization. Pasteurization can be batch or it can be flash. The next one is the ultraviolet or ionizing radiation. UV variation or UV irradiation is considered to be alternative to traditionally used thermal methods like pasteurization. Initially, these UV treatments were applicable only to solid foods and not to liquid. But as advancements continue, we now have UV radiation being applied to other liquid foods and even beverages. This process is very, very important for the liquid foods, and the properties of the food also remain intact. The next method is the new methods now that are emerging that are non-thermal treatments. These are new non-thermal treatments that are used for preserving food. The first of them we're talking about is the high hydrostatic pressure, followed by the pulsed electric fields, oscillating magnetic fields, and photodynamic effects. The high pressure processing method is also known as pascalization. It inactivates harmful pathogens and vegetative spoilage microorganisms by using pressure rather than heat to effect pasteurization. It promises the same level of food safety and heat pasteurization as pasteurization, but without damaging nutrients in the fruits and the vegetables. Usually heat damages, but in this case, it uses pressure rather than heat. Several studies have found that even vitamin C is relatively unaffected by this process. It's a method of preservation and sterilization of food whereby the product is processed under very high pressure, leading to the inactivation of the organisms and also the enzymes of the organisms in that food. It has limited effect on the covalent bonds within the food product. That is why sensory or nutritional aspects of the food is retained. During this pascalization, a lot goes into the food. By preservation, the pressure goes into the food, makes the food to be intact, and prevents entry or infestation of microbial agents. HPP can pasteurize food products with decreased processing time, reduced energy usage, and also less waste is generated. Another one that is a non-thermal method that is emerging now is the pulsed electric fields. This one uses short pulses of electricity for microbial inactivation and causes minimal detrimental effects also on the food quality attributes. Therefore, it also provides high quality foods. PEF technology has a wide range of applications. It's used for liquid foods, semi-solid foods, and also solid foods. It uses the induction of electrical field and electromechanical compression that will lead to the pore formation and instability in microbial membrane. The next one is the oscillating magnetic fields. This preserves food by sealing the food in a plastic bag and subjecting it to several pulses in an OMF at varying temperature for specific total exposure time. The ultraviolet radiation in its own method uses wavelengths. The UV light occupies a wide wavelength range in the non-ionizing region of the electromagnetic spectrum between X-rays, which is 100 nanometer, and visible light, which is 400 nanometer. The wavelength of UV light is usually divided into three bands: the UVA, UVB, and the UVC. The lethal effect, that is the damaging effects of the UVC, and its effectiveness as a disinfectant are already known to us. However, microbial responses within food to this emerging technology is recently being investigated. The shortwave ultraviolet processes, when it's applied to heat-sensitive fruits and vegetable juices, they are an interesting preservation alternative because important microbial reductions are generated without meaningful destruction of sensory changes, without also destroying the shelf life, and make the food acceptable. UV light has advantages that, number one, it does not generate byproducts, and then it does not also generate chemical residues. It's a dry process, it's simple and it's effective, and it's also economical. The technique of the photodynamic inactivation is also a new and promising strategy. This technique is based on the use of photosensitizers, which are activated by appropriate wavelength light. Several light sources such as laser emitting diodes and halogen lamps are currently used. The light emitting diodes have been used as alternative light sources because they are cheaper. They have wider emission bands, they are easy to use, and also they have greater flexibility in irradiation time. The toxicity of this PDI method is in the fact that it produces what we call ROS, reactive oxygen species. That is toxic to microbial cells. It leads to the cell death. They are also known as free radicals. They hinder microbial resistance, and such the microorganisms are not able to resist the ROS because they are oxidative in nature and they destroy the microbial cells instantly. For instance, the other organic structures like the lipid membranes, the proteins, and the nucleic acids are destroyed as a result of the oxidative nature of the ROS. The advantage of this photodynamic inactivation is that toxic chemicals are not also generated, and the only energy that it requires is the light source. There is also low probability of triggering the development of any resistance by any of the organisms, so it can be used for several foods. With this, I would come to the end of the physical method of food preservation. The next lecture, I'll be looking at the biological preservation, and then I'll have a final lecture on fermentation of certain foods and also some quality assessments that I would like us to know. Thank you very much. Have a good day.